Okay, hello everyone. I'm glad to see you at the coffee break, this TRANSFAC. Actually, we have a lot to do today, so there were a number of questions before which we collected together. It's all about basically motifs and de novo motifs, I would say. And that's a very interesting topic, and I decided to have a bit of our time devoted to that topic about motif in general, and especially what can be done with de novo motifs, what they are good for, why should we find them, and how can we use them. So let me share the screen. And you see already some formulas, but don't be afraid. Actually, I would like to go back to really some basics, I would say. So I would now start this presentation, which I've prepared. You see on my slides, there are some motifs. You call them motifs. These nice pictures with T and C and Gs, and some of them are bigger, some of them are smaller. Why is that? And there is certain misunderstanding sometimes what people actually call motifs. And in fact, good collection of motifs is a TRANSFAC database, which is, you can see, for instance, in the free version of it in this view, or you can download it if you have a license. And then you see this like 4 nucleotides and the positions of the site where the transcription factor has to bind. And then basically this is a so-called positional weight matrix. And in fact, positional weight matrix is a motif. So, but some people call motif something else. Let's first understand what it is. Actually, as in music, what is a motif? Motif is some kind of pattern in the music, I would say, or in any kind of a consequent events, which repeatedly happening in that music, for instance. So then we call it a motif. So that music has a certain motif. But of course, if this pattern is always the same, it's a very simple motif. It's just a repetition of the same consequence of sounds. But very often in very complex, complicated music, you can see a motif with a certain variability, right? So you see the variability of your sounds, but you still can recognize that motif. And in that case, motif is some kind of a model which describes a similar consequence of nodes. The same here in nucleotides. So basically, in the nucleotide sequence, in DNA, there are certain places in DNA which can function in a certain way. For instance, they can be sites for binding of a transcription factor of a certain type. And then these regions are similar to each other, all right? And then some people call each of that region as a motif, but I would say it's a wrong definition. In fact, the motif is a model, is a sort of a model which can describe all those repeating parts which you observe in the sequence. So in our terminology, we usually say each individual region is called a site. So basically site, a DNA site or transcription factor binding site. Like in this case, you see each of that nucleotide sequence has these capital letters which define that position where where transcription factor mainly binding to DNA, and we call this sequence of nucleotide a site, right? But then each site may be different from each other, but they have some similarity to each other. And to define a motif which describe all those sites, in our terminology, we call it a site model. So sometimes we call it a motif, sometimes we call it a site model. But a particular representation of that site model or particular type of a model is a model described by positional weight matrix, right? So when what we do, we align all these sites to each other, putting similar positions one after under another, and then we count in how many times you find in that position T or G or C, and we put this in such a matrix. And then this matrix actually describes in some way all these sites together. Interesting that, in fact, it describes all the observed sites, but in fact, it also describes all new sites. So, in fact, with the same matrix, you can try to recognize the beauty to make some in the TRANSFAC, which is called match, which takes such a matrix and scans it through the sequence which you want to analyze. And in each position of that sequence, it computes by going through that matrix sort of a sum of those contributions of each nucleotide, and then it calculates a score. And if the score is higher than a certain threshold, then it defines its motif. And in fact, that's actually correspond to a certain physical process which is happening when transcription factors are binding to DNA. And as was described early in the history, well, some years ago, in a very nice work of Berk and von Hippel, where they actually put a very basic biophysical background under this positional weight matrix approach. So you can find these papers and read about them. And basically, they based the calculation of their description of that process on several assumptions. And one assumption is very important, that actually that each position in the site contributes independently in the energy of binding of that transcription factor to that place, which of course a very simple explanation, but it works in most of the cases. So we know that in reality, the positions of the sites are in fact correlating to each other, so they can influence what nucleotide is staying in the first position, influencing on what nucleotide can stay on the second position. That so-called Markovness is present in the real sites, but the very basic model assumes that each positions contribute to the energy of binding independently. And there are several other sort of assumptions here, but all in all, they describe the distribution of transcription factors when they bind to their target sites pretty well. So they basically build those formulas according to Bernoulli law, and they found out that in fact, if you calculate the energy of binding according to this even contribution of each nucleotide, or not even, but independent contribution of each nucleotide in energy, you describe it pretty well. So, but I don't want to really going into that details. The only message I want to give you now that the motifs, so this word motifs, is in fact very complicated, and there can be several or many different models how to describe similarity between observed binding sites for the same transcription factor. And that depends on how we build the model, which assumptions we put in the model, for instance, independence on the positions. Actually, it also depends from which data we are learning, we are building such a model. And I will skip those big formulas, but let me just go to several main pictures at the end. So at the end, when you build the model for this, for instance, you build a positional weight matrix for a given transcription factor, like in this case, we build it for E to F transcription factors, and we will talk about them today. So we have built them on a particular set of known sites. So you see, this is actually a collection of factors which were used to build such a matrix. And that's another actually problem which you see here. We were talking about that before, but let me briefly repeat here because maybe not everyone were previously on my on our discussions. So as you can see here, we put several transcription factors, not just one, but several from a big family of them. You see here factors E2F1, E2F3, E2F5 from different species, from human, mouse, actually E2F4. So why is that? So the reason is that actually all these different transcription factors, different proteins, they function differently, but their DNA binding domain is pretty similar, almost identical. And in case when you don't have enough sites for one particular transcription factor, but not only in that case, also in case if you really want to understand and build the model for a whole family of factors, to be this model more kind of sensitive to any new family, perhaps, which we don't know yet, or to basically open a possibility to be more sensitive in predicting new sites. Then you are collecting all the sites for different factors in one group, and then you build this model based on those sites only. So in case if you have enough sites, and you can build such a model just from sites of one particular factor, you also can do it, and then you will be building a more specific one. So that's another variability in that model construction, in the construction of the motifs. That's why, as you can imagine, when you do all these various ways of building your positional weight matrices, depending on how you collect your factors together, depending on which site collection you are using to build this model, depending on what kind of assumptions you put in building this model, whether you consider positions independently or you consider them correlated to each other, you can build a lot of different models, basically for the same factor or for the same family of factors. And somebody is asking why TRANSFAC, for instance, has so many motifs of so many positional weight matrices collected for one particular factor. So if we will see today that, for instance, for E2F1, there are 33 positional weight matrices collected in TRANSFAC, and they are all different. And which one is true? All of them are true, because actually, all of them were built on certain assumptions, on a certain collections of binding sites, and they describe the true nature simply from different angles. And now this question comes. We have already 33 different motifs for E2F. Do we need more? And now we are coming to this question of searching for de novo motifs. And that's really the true question. When and why do we need new motifs? But first, let me explain. Okay, so maybe these different cutoffs will come to that a bit later. But what I want to show to you, these algorithms for de novo discovery, de novo motif discovery. So very early in this whole world of DNA motifs, it stands this question that how to find new motifs in a set of sequences, especially if the set of sequences is not really restricted to that region where transcription factor is indeed binding. It's easy to build such a model, like a positional weight matrix, when your sites are really restricted to a short region, like it was done in this case. So in different papers, and very early in the history of transcription factor omics, we can call it, people were doing very careful experiments. So they were kind of, searching for the regions of DNA where the factor is binding. They were cutting the flanks, which are kind of extending out of the region which factor is covering by different restriction enzymes. So they were trying to really minimize the region and understand where the binding of the factor really happening. And then in such a case, the list of sequences which you have to align and to build your positional weight matrix is very small, very easy. So they are very short. There are some very simple algorithms which can quickly align those sequences. The problem comes when there are actually regions which comes from high throughput experiments. For instance, the regions which coming from ChIPMunk or ATAC-seq. So in the difference, that the ChIPMunk classically is done by a certain antibody to a certain transcription factor or a family of factors. And then in advance that those regions of DNA which you sequenced, they might be of different lengths, but these lengths could be quite big, up to 1,000 nucleotides, or like 100 nucleotides, 200 nucleotides. There are new methods which can make it much shorter, but generally you have those regions which are supposed to be bound by this factor pretty big. And we know, since it was done by using the antibody to a particular factor, we know that that particular factor is binding to that region. So these regions are somehow bound by this factor. But there are other methods, for instance, method called ATAC-seq, when it searches for just open chromatin in DNA. And in such a case, actually, we know that region is active, it's opened, but we may not know in advance which transcription factor is actually binding to it. So this method just gives you, that's the region which is active. So at the end, we can have a list of regions from DNA which are functioning in a certain way. And then the question stands how to find a motif, or maybe few motifs, several motifs, which are, which are overrepresented, or which are abundantly present, or which are dominating in that list of regions. And that's kind of a difficult question, because those, as I said, those regions can be very long, and those motifs are usually quite small. And where is this motif? You don't know in advance. And very early, it was proposed quite interesting method in general, which has two steps. In statistics, it's called estimation and maximization steps, which goes in circle. So basically, at the first step, you make some assumption about the motif. It may be a very random assumption. You can say, I don't know the motif at all, and just put some random numbers in this table. And then that's my motif, which probably is there. And then I start searching for the sites using this motif. And then it will find some sites, even if the motif is random. And then I will align those sites, and then basically build the better representation of the model using these aligned sites. And not only, it's actually, well, when you're doing aligning, I can also somewhat shorten or increase the size of that motif. In fact, I can shift a little bit left and right, so I can adjust this original motif a bit to reality what I see. And then I repeat this iteration. Now I use the new motif. Again, I search a new positions of it. and I align it, and I do iteration. And as you can see, this algorithm is pretty sensitive. As soon as it can see some repeatedness in that sequences, it's very quickly converges to the model which it sees. In fact, it's very similar to how our perception is working. As soon as we see something, our perception is actually focusing on that object, and now we see it more clearly. And also, if you are in the dark, you can find some objects which are not really there. So your perception can create something phantomly. That happens with you sometimes when you in wild somewhere in a forest and in the dark night, and you can see some very strange faces looking at you from the forest, right? So your imagination actually creating a certain pattern, certain motifs which are not really there. That may happen also with that algorithm. So it may come up with a motif which is not really there because it's started from something which was not really true, and then it suddenly converges to something which may happen there from time to time, but not really abundant. So that's why there are some advantages, some new features in that algorithms. There are new algorithms coming which would start not from the random motif, but the motif which you already you can put some prior knowledge about that motif, or it can do some tricks to get this initial motif in your hands. And one of the best algorithm which was created several years ago is so-called algorithm called ChIPMunk. It was done by some of my colleagues. We can actually, this is a paper. This is one paper where they actually applied that algorithm, and that's the paper which describes that algorithm. It's called ChIPMunk. Yeah, I can now maybe open it again. So if you look for ChIPMunk, but of course, if you just search for ChIPMunk, you'll find a very nice animal, but you have to put motif and in DNA, some other keywords to get to that tool. So this is a website where you can get this tool, the original R code, and actually not R, but the source code, and you can install it on your computer and run it in the full functionality. So we made our efforts to put this algorithm into our tool, and let me quickly show you how this tool works, so that you feel actually how these motifs can be found in your sequences. So okay, so let me go to our website. And by the way, don't be shy, just put your questions in Q&A. So they will be collected there. So at some point I will stop my talk, and I will come to those questions, and who will be the first that will get the first answer. So let's go to our GenExplain website. And we go now to another. If you remember, previously I showed on our main server GenExplain platform. Now we will go to our secret server called Genome Enhancer, and that's another topic about Genome Enhancer. I will keep it for several series of our seminars. But now I will simply use this server to demonstrate this ChIPMunk algorithm for you. So to do that, I have to actually go to the platform view from that Genome Enhancer server. And now you're at the GenExplain platform. So for those of you who didn't have access to it yet, just get access. It has a free accounts which you can open very easily, and then you can explore this this platform. Of course, when you have the full access to the, so you have a full access to the platform when you get a free account, but of course the databases, commercial databases like TRANSFAC are not included in the free access. You have to add it when you have a license, but generally it has a full functionality. So basically, for instance, this ChIPMunk can be used under free account as soon as your data are enough. So okay, here we go now to my projects, and here I created a new project where I can put my examples. So let's create here in my project a new folder. We will call it demo. So it's demo 1 or demo motif. So let's create such a folder where we'll put our data first. So what kind of data we are going to have? As you know, as I said, this the first important question about de novo motifs comes when we start analyzing ChIP-seq data. And again, so why, for instance, we have a lot of motifs for transcription factors already in TRANSFAC. Why do we need a new motif? And the reason is sometimes we need it because there are special, factors are so tricky. So in different conditions, they may bind to their target sites in different manner. So, if you do your experiments in very special conditions, which were not done before, there's no ChIP-seq data available yet done in the conditions you are doing right now, under a certain treatment, for instance, by a certain drug or whatever. So the transcription factor of your focus, maybe it's a general transcription factor like E2F1, but it can have a certain modification. It can have a certain cofactors which modifies its mode of action, and it starts binding to slightly different motifs or sites, and consequently it has a slightly different model of binding from those which we already collected in the database. So that's why you might be interested to create such a new motif for the same factor. That's one case which can happen quite often. Another case, if you are coming from a completely different domain, so you are studying some new organism or some new genome, and you don't know about where transcription factors are not well studied yet, as in human, mouse, or rat. So you're coming from a genome from some plant or fungi. And then, of course, this de novo motif search can be very helpful because you basically don't know much about any transcription factors which are actually working in your cell. So, for instance, you have your transcription factor, you made a ChIP-seq experiment with the antibodies to that factor in that plant or in that new organism, and then you find these binding regions in DNA, and now you need to find the motif. You need to actually to come up with a logo plot to show in your paper that this factor is actually binding to that, it has this motif to bind. Okay, so for that we will go. We need ChIP-seq data. So ChIP-seq data are available in various databases. For instance, you can get it from TRANSFAC. I also want to show you in this nice database which we published some time ago together with my colleagues. It's called GTRD. So you simply Google it. It's GTRD, Gene Transcription Regulation Database. Is actually one of the biggest repository of various ChIP-seq data and also toxic data now. So, and it actually gives quite easy way to get ChIP-seq data in bulk for a given transcription factor. For that, you simply go there, and it has a simple interface. You can search for your factor here. You say E to F1, so it tells you we have that, and then you search it, and then it gives you here below a list of those experiments with E to F factors. And you see there is not one experiment. There are many experiments, and they are done in different conditions, in different cell lines, in different treatment, in different actually species. And you simply select the one you need, or here. So I forgot which one I selected, but just to show you, you selected, for instance, this one, and gives you information about which was where it was done and what was the conditions. And here you have an export button, so you can export this whole data in either in a FASTA format or as a format as so-called intervals or BED format. And here you can save it, and you will have it in your computer. But now in the geneXplain platform, we have to upload this BED formatted pics from that experiment. So we have to click here to import, and we go to our computer, and here we actually. So I choose this one because I imported it before. So these are the intervals from this one of the E2F1 ChIPMunk experiments. So I'm importing them now into the system. So it asks me which kind of genome build it is using. So you have to know that. So in this case, I know that was actually the latest genome, HG38, a human genome. And then we simply start an import. And it shows me directly those regions on the genome browser. And you can also see them by clicking on the right button as a table here. And you can see there are 29,000 ChIPSeq regions over different lengths. And you see, if you sort them by lengths, they vary from 140 to actually 1,600, so pretty much variable. And here you see also some values associated with those regions. So the probability, the p-values, and so on. So we have now those peaks, and they are actually distributed among the whole genome. So let's have a look. Let's have a focus. Again, the focus. Always you have to make a first focus on something. So let's make a focus on those sites, those regions of E2F binding, which are located near start of transcription. We know that E2F is actually acting very near start of transcription, and it may bind also in other places, but most of the functionality when it binds to the near TSS. So how to find which of those peaks are actually binding near start of transcription? So the first thing what we do, we actually just put them on genomes in genes in general. So for that we have various methods, and we have here the method called then the number of methods. Let me check how we can find that. It's called track to genes. Track to gene set, right? So we take this track and we simply put it here, and we specify what regions of genes we would like to see, and we will see 1,000 nucleotide upstream of TSS and 1,000 nucleotide downstream from 3' end of the gene. So including the whole body of the gene together with 5' and 3'. And we want to output everything, what it can output. And so we can say it's a track genes for E2F1. So then we run it. So now it's start mapping of all these all these peaks on the genome and comparing them with a gene annotation. And it gives such a nice table, which actually, you see this 20, how many was there? 29,000 peaks. They actually mapped on 20,500 genes, basically practically all genes in genome. But they are mapping to different places. You see this, each gene is represented by this nice picture. These blue boxes are exons and introns are these kind of curves. And of course, the size of introns is not really proportional here on this picture, but just demonstrating. And this red bar is showing where the ChIPMunk peak is located in that gene. So what we are interested in, actually, those peaks which were found in 5' regions of the gene. We sort this table by the 5', and you see these are the genes, and these are the peaks which are located in mainly in 5', but there are also some in introns. But what we need, we just need these 5' gene, 5' located peaks, how to extract them. So to do that, we have some other tools to do. So, but let's first filter this list of genes and select those genes which have at least 2 peaks in the 5' region. Okay, so we say above threshold in the field, 5', and it should be higher than 1. So 2 or 3 or more peaks inside the promoter. So actually, this 5' is describing the more or less a promoter region of the gene. And we see there are 956 genes where the peaks, more than 2 peaks, are located in the promoter regions of them. We can save this list of peaks. We can say E2F1 site in promoters. And with that, we see them then as a separate table. So if you have questions, just put them in QA. To what I'm showing also, maybe I can also interrupt if there are some urgent questions about during my show. So now we see these 956 genes in a separate set. And now what we want to do, we want to actually have a set of regions which correspond to the promoters of those genes. For that, we have another program which is called gene set to track, to promoter track actually. Let me run it. So what I will do now, I will go to this folder. So I will take this table, put it here. This is the table of the genes where we have peaks in the promoter, and we want to have the regions, these promoter regions in our hands. So, and we want to have them minus 1,000 to 100, and we run them. So it will create to us now actually these promoter regions. Just to show you what actually we have now. So we have, if you put here the genes on the genome browser, so and zoom it a bit out, what you see here, the genes, and you see here actually the promoter regions of those genes of 1,000 nucleotide upstream and downstream. And these are not just genes, any genes. These are genes where our tracks were located. So if you put here the tracks, you can see them. So they are actually located in some of them located in this promoter region. Some of them are located outside. But as we thought, we told, we want to really focus on those tracks which are located in the promoter regions only. So we want to filter these tracks, all these 29,000 tracks. We want to filter and extract only those tracks, those peaks. I'm sorry, which are which are located in the promoters of the genes. So, and now it's very easy. So we have the track for the promoter. We have the track for all the peaks. and we have to intersect them. So for that, we have a function which is called this filter one track by another track. Where we take this track of 29,000 peaks, and then we take a track of our promoter, and we say that the distance between the beginnings of those peaks has to be at least the size of the promoter, which is 1,100. And now we can run and get basically a new track, which has just 1,888 peaks, and this is a track which contains just these peaks, including which are basically intersect with the promoter. So we can see it again if we put this track here. We see this contains only those peaks which intersect with the promoter. Okay, so now we have this track, and finally, I'm sorry for such a long introduction, but finally we can apply the motif search, the novo motif search for such a track. So we go now to our toolbox, and here in NGS, for some reason we put it in NGS category. We have this ChIPMunk program, but building even more complicated site models, which is not just mononucleotide positional rate matrices, but dinucleotide positional rate matrices. You can read about them. Maybe we can have another session about these more complex site models. But for now, we will use just a standard ChIPMunk to find my motifs. It's here. So we take our list of peaks. And now what we do, we have to specify a little bit our parameters. We can, of course, use them as they are by default. But you know, the start lengths, the stop lengths, actually it assumes that the site can be very long, up to 16 nucleotides length. But we know E2F is a very small site, so we can easily say its maximum should be 10, and minimum, okay, it should be, for instance, 7. Okay, and then motif shape, it also influencing somehow on how motif will look like, whether it's kind of flat, it can be, it can search for motifs which have a conserved nucleotides everywhere, or it should be a single, which means it searches for such a very concise motif which has most of the conserved nucleotides in the center of the motif and the flanks which are not so conserved. So kind of classical motif. And we can, it will output in the library, it's called matrix library, and the name of the matrix, let's call it E2F1 new. So we will build a new matrix. So now it's start basically building such a matrix. It may take a little bit of time. I see already questions. Maybe I will go. Maybe there are some questions concerning. All right. So the first question. I'm sorry. I'm a bit confused. Is there input file a narrow peak file? It can be narrow peak file and can be just a standard peak file. So in this case, we took a standard size peaks. And actually, I'm not quite sure what you mean. What do you mean on the narrow peak file? So there are, it's very interesting. There with a time and a new kind of terminology coming up. Yes, I understand what you mean. So basically, there are some peak calling algorithms which are trying to build so-called narrow peaks using some types of assumptions, and they can do it. And in such a case, of course, for you, it would be better to use such a narrow peak. So narrow peaks means they try to really minimize the size of the peaks to the minimum as much as possible. But of course, that depends very much on the depth of the sequencing. As you know, if your sequencing is very deep, you can really see clearly which regions are occupied very frequently by the binding of transcription factor, and you can build such a narrow peaks. But if your sequencing depth is not very high, then you have to you have to work with a longer peaks. So if you have narrow peaks, you can put them. If you don't, you can use what you have. TRANSFAC. So there is other questions concerning TRANSFAC. I can, okay, maybe I misunderstood something. It's called, can I perform TFBS search with TRANSFAC alone? Yes, and that's what I was talking before in my previous coffee breaks, where I just used TRANSFAC alone without any new motifs. Today, I decided to describe how to come with new motifs. If you want to see how to use it just using TRANSFAC, please go to our website, find the recording of our webinars. Well, it's not webinars, these coffee breaks, and there are some answers. Actually, we can repeat it, and some of that I will show a bit also today if time permits. Thank you for that question anyway. And there is another question about mutations. Let's keep it. I will answer when I finish with those motifs. Okay, thank you for time being. Okay, so as you can see, it found such a motif, which it claims it's actually a motif for E2F1 transcription factor. So CC, G, CC, very symmetric, I would say, or kind of symmetric. It's a little bit different from the standard classical E2F motif. Just very briefly, let me go to show you the standard motifs. If I have them here. Okay, maybe we can just go quickly to our website. Also to show you a bit how to find, to navigate in TRANSFAC. So now we go to the TRANSFAC itself. So the database which collects all those motifs from various literature. And here, yeah, we can go to TRANSFAC or to a general database which include everything, not only TRANSFAC, but also pathways and diseases and biomarkers. But now we are focusing on TRANSFAC, and we search for E2F1. So we search for E2F1, and you see 4 different species, and we go for one which is from human. And it has a lot of information about this gene and protein, how it is associated with the diseases, and which tissues is expressed, and how the gene for E2F factor regulated by itself, regulated by other factors. But what we are interested now are looking for the motifs. And you can see there are, I said 33, no, 32 motifs. And as you can see, these are the motifs for which are documented for E2F from various literature, or we were building them by ourselves, or they built from the various high throughput experiments. And as you can see, it is, yeah, this one is somewhat reminding what we found. You see GG, CGG, well, a little bit different. But most of the case, you have either track of A's or track of T's, which we don't have here so much. With a little bit of T's, but quite rare. So which makes a bit strange. But on another side, as I said, maybe in that particular situation, the TRANSFAC is, this transcription factor, E2F transcription factors, tending to bind to such kind of motifs. Okay, let me quickly show you what we do next. Next, we want to use this motif to find these binding sites. and in any other regions, right? So, and it's very easy. For that, we have to take this library, so this matrix library, so which has just one matrix. It can have several matrices. For instance, if we run not ChIPMunk, but ChIPHort, which actually gives you not just the one best matrix, but several also suboptimal ones. But in this case, it just gives one. And then what we do next, we go to site analysis section here, and we build a profile using this matrix. Why do we need a profile? Just let me build it first, and then I will explain what do I mean. So the matrix itself is just actually a model. But you have to apply this model with a certain cutoffs, with a certain parameters. And to estimate those parameters, and that comes to the figure which I didn't explain to you, but maybe you remember these curves which I showed in one of the slides. They helps you to find the optimum parameter, what cutoff has to be chosen to find the true sites for this matrix. And for that, we have various templates for different p-values. So which means if you select 0.0.1, it will find such a cutoff, which sort of guarantees you that these sites will be found approximately one site per 1,000 nucleotides in a random genome. And as a random genome, we use a human genome. And then it will create such a profile, which is such a file which has a matrix itself, and it has actually the cutoff 0.83, which gives you the, as I said, the guarantees that you find one site per approximately 1,000 nucleotides. And now you can apply this profile, this matrix, to search in your sequence. For instance, in any sequence. For instance, in the same sequence of those motifs, of those tracks, which we were just analyzing. And for that, now we go to the program called site search on track. And we put actually the track where we want to search our sites. So you remember there was this promoter track. Where is now promoter track? Yeah, this is our promoter track. You remember 1,888 promoters. So we put them here. And then as a profile, as you can see by default, here is a TRANSFAC profile. And that's an answer to the question we had just had. So if just forget about any other motifs, search for the motif, anything, I won't just use TRANSFAC. Here you are, it's default. So by default, you are using a profile from TRANSFAC, which is called vertebrate non-redundant mean sum. So it's a selection of best matrices from TRANSFAC. But now we want to use actually not the TRANSFAC motifs, but the motif we just created. So we put it here. And we just name the output. We create, so it's E2F new. And we run it. So it takes these motifs and it put them, finds the sites. Here they are. So let's put them on our genome browser. Just here. So these are the sites which were just found. You see, all right, okay. So we have actually found the site exactly in this peak. And here you can click on this site and you see again the log plot of that motif, the actual score of that site. So it's very highly scoring. So we go for the next site, we see find it here and so on. By the way, I also applied to these tracks the TRANSFAC and all the matrices, E2F matrices from TRANSFAC and I found them here. So if we drag and drop them here, you see them here. So what I did, I just applied not the new profile, but the profile from the TRANSFAC. And here you see that actually our new motif found in some places where the existing motif from TRANSFAC also found a site. But actually this region obviously very heavily occupied by various motifs, various sites similar to E2F binding motif in general. So you see a lot of sites, and to demonstrate how many of them are there, that's kind of goes already beyond the talk today, and I will talk about maybe next time about that. So what we can do, we can actually search for frequencies, how many sites found in all these peaks, whether these are, when can we compare number of those sites found here is a number of sites found in completely random regions, and can we find actually enrichment of the sites. This is done by special programs in our platform, and I just want to show you output. It's a summary output. It's a simple table. It compares the frequency of each motif of each matrix. So frequency of sites found by each of that matrix in the peaks from ChIPSeq for E2F, so which means approximately 10 sites per 1,000 nucleotide. And here, how many of those sites are found in randomly generated regions in genome, so just randomly picked up from genome. And it's approximately 1.4. And enrichment score is here 7 times. And actually, the biggest enrichment score is given by this very general matrix, which basically I showed at the very beginning, which includes not just E2F1, but sites from various different factors. And it was built maybe 20 years ago from very little knowledge about those sites. But as you can see, it gives now the biggest enrichment. And that's why you never throw away old stuff. Sometimes it may be useful. And if we compare, and you see the enrichment is 15 times, and the p-value is actually beyond any significance. It's it's 0. But interesting, if we take this newly created motif on the same set of peaks and look at the statistic of that motif, we see also quite good enrichment. It's 12. It's nearly the same as here. Yeah, show it here. So here you see the biggest was 15, the next one is 13, the next one is 11, and the one which we just built is 12, which means, of course, you can say it like it was built on the same set of sequences, and now we are looking at this set. It's kind of effectively a training set. But in another way, we can say probably really by doing this de novo motif search, we find a kind of a new motif, which is specific for those type of experiments in that particular cell line, in that particular conditions. It was actually done in pancreas cancer, as I remember. I was reading that paper, and in that particular condition, this factor maybe was probably binding to a very specific type of the motifs. Okay, so I finished with my motif answers, and I'm sorry I just took the whole hour, but I have time. If you have time, stay with me. I will go now with the answers. I will make a first little pause just for you to put some more questions if you like. And by the way, for those of you who are not now, but in future looking at this video in our YouTube, it's the right time now to put a like in our YouTube channel. Okay, before any other new questions coming, let me answer this one which we have now. So can geneXplain find mutation in the intron when compare with reference sequence, for instance, HG38. Okay. All right, so I can answer this question. It's somewhat completely different from the topic today, but that's really good because it opens new opportunities for you to listen something new. So, as you know, this question is very basic question. Just to repeat it. So, if someone has a sequence, he sequenced a new genome, or for instance, a tumor genome, tumor-derived genome from human, and he wants to compare it to the reference genome and find all mutations. And that's a classical task for bioinformaticians, and that's nothing special for GeneXplain. So many bioinformatics tools can do that, but I must say we want to help everyone who comes to our platform, and we have tools of a general use. So as you can see here, we have a Galaxy incorporated in our platform, and the Galaxy, for those who may not know, it's quite a good tool. You can also find it online in various servers. You can install it locally. It collects a lot of functions, a lot of tools for analysis of high throughput data, for analysis of genome sequences of RNA-seq, and we also using it in our pipelines to analyze data if users are coming not just with ready sequence, but with raw data. And what I mean raw data? Raw data means, for instance, raw data coming directly from a sequencer. So as you can see here in our home page, here the home start page of our tool, if you have a genomic sequence, then you can actually, you provide here various pipelines how to process the genomic sequence and find your mutations, for instance. We have this section called genomic variants, and for instance, here find genomic variants and indels from full genome from HG38. So if you click here, it gives you actually the whole pipeline. It's just not just one function, but it's a pipeline which you can see here. What it does, it first take your file, and in this case, it takes FASTQ files. So it assumes you are doing your sequencing using, for instance, Illumina machine, and you come up with FASTQ files, and you can sequence either. So in this case, it assumes you are doing the most newest way by doing forward and reversed. So this you get actually two FASTQ files for one sequence, and you put them here, and then it first map them on the human genome, and then it will mark duplicates. It goes again to realign, doing a lot of actually quality testing, and at the end it outputs the mutations in a so-called VCF form. So a VCF is a standard format for mutations. It output them and it visualizes them directly on genome browser, and you directly see those mutations. That's one thing. Moreover, you can proceed further with analysis of those mutations, and that's something that goes now beyond your question, but probably you might be interested in that. As you can see here, once you found your mutation, so what? So what to do with them? And then what you can do, for instance, you can search for transcription factor binding sites which are affected by this mutation. And that's very important part of our tools. And I must say it's pretty unique. There are not so many tools available, even free tools or commercial ones, which So quickly from mutations, you can go directly to predict which binding sites are affected, whether this is affecting a lot of sites for the same factor, whether these transcription factors are really playing a particular role in certain pathways. So this all can be answered in that section of this of that tool. So, but that's as I say, goes a little bit beyond what we were talking today. But I think we will define a certain coffee break to answer these type of questions about how you can analyze your mutations. You know, mutations can happen in various places of genes. They, of course, many focus is currently done on mutations which are affecting the proteins. But in our tool, you can really analyze mutations which are affecting the regulation of genes because they are affecting the binding sites of transcription factors. Okay. Okay, there are some questions also in chat. Yeah, so the people ask saying that I think SNP analyzer is what I need. So that's what I'm saying. So okay, no, that's I'm sorry, that's a different thing. So you again the question was, I think the SNP analyzer is what I need. So that's something different. So basically, what I was talking about right now is mutations, for instance, happening in the tumor in the somatic genome. So you get the DNA sequence from that genome, and you compare it to the reference genome, you find all the mutations. But what the question was about SNPs, and SNPs are these are to be really good in terminology. These are not mutations because SNPs are single nucleotide polymorphisms. Polymorphism means they actually happening in a normal population. So people carrying those polymorphisms, they are not mutant, right? So people are not mutant, they are normal. So that's why they are not called mutations, but rather polymorphisms. And those polymorphisms, they are detected by various tools, and they are put in the biggest databases, so-called DPSNPs and other databases. And each of that polymorphism has a certain accession number, so-called RS accession number. And our tool can also work with polymorphisms. Oops, I think it's here. Yes, you see, it's called SIFT, for instance, is a tool which takes a list of SNPs. It's a very simple list. It's a list of RS numbers, nothing else you need. And then it maps them to genome first, and then again, it starts from that. You will visualize it on genome. You will then start analyzing how this polymorphism can affect binding sites of transcription factors or the binding sites around the SNPs. That's the whole new area, which I would be glad to describe, I guess, in the next session. And the answer, yes, I need to analyze the variant of certain significance in the intron of one particular gene. I do a gene. Introns don't have RS. Okay, so I think I don't understand what do you mean they don't have RS, but maybe you can send a bit more detailed question. You can send it also by email. So I will try to answer these questions directly to you. Okay, reference sequence RS. Okay, I understand. Okay, maybe next time I will put more attention to that particular type of question. All right. Okay, any new questions? We still have some time. Maybe you have questions, just the general questions about our products. Probably not. Or our tools. Just don't be shy. Put more questions. Okay, so I think you are exhausted so far, as well as me. You know, I'm really glad that I was able to talk, because just 2 days ago I got corona and I'm now fine. But it seems I'm over using my good drink. Okay, I think we can finish for today. And thank you for your questions and for your attention. Welcome back next time. So we are doing these coffee breaks pretty regularly, approximately 1 to 2 weeks. It can deviate depending on various events, but basically we try to keep this and also send us the questions in between by emails or by special form, which you've seen on our website, so that I will prepare them in advance and give the best answers for you. Thank you very much again, and see you next time.